Substances such as oxygen, carbon dioxide, water, dissolved food molecules, mineral ions, and urea need to be transported into and out of organisms, which usually happens through diffusion. And the reason why these substances need to be transported into and out of organisms are due to various reasons, such as oxygen and glucose needs to be diffused into cells for them to do respiration. You also need to diffuse out waste products such as carbon dioxide or even urea if it's a liver cell. Unicellular organisms can diffuse enough substances in and out of cells because of the fact that they have a very large surface area to volume ratio. This is because they are very small in terms of cell size, and that makes it easy for them to diffuse enough oxygen, water and food molecules into the cell and waste materials such as carbon dioxide out of the cell due to the large surface area to volume ratio. Why are humans not made of one extremely large cell? The larger a cell surface, the more substance that you can diffuse into and out of the cell. But the problem is, if a cell volume becomes too large, the cell can't fill up with all the material it needs for metabolism. So, in other words, as the cell becomes larger, although the surface area increases, it can't get enough of the material to do reactions such as respiration for the size of the volume it's now become. Now the surface area to volume ratio is calculated by surface area divided by volume. So here we've got an example here of three different types of cells. You've got a small, medium and large cell with different dimensions. And to work out the surface area, because this is a cube, that means it's 6 times 1 times 1. And then for the 2 times 2 times 2 block, it's 6 times 2 times 2, which is 24. And then we've got 6 times 4 times 4, 96. So in other words, to work out the surface area, you just find out the area and you multiply it by how many different surfaces there are, in which in this case there are 6 different surfaces. And then to work out the volume, you just multiply all 3 different dimensions, so height, width and length. So 1 times 1 times 1, 2 times 2 times 2, which is 8, and then 4 times 4 times 4, which is 64. And then to calculate surface area to volume ratio is surface area divided by volume, so that's 6 divided by 1, which is 6, 24 divided by 8, which is 3, and 96 divided by 64, which is 1.5. In your exam, you might be asked to calculate surface area to volume ratio, but it might not be a cube. Okay, so when we analyze this, we can see that as the cell increases in size, the surface area to volume ratio decreases. So as organisms get bigger, the surface area to volume ratio actually gets smaller. So both surface area and volume increases, but volume actually increases at a faster rate than surface area. And this means that you can't rely on diffusion. You can't diffuse in enough material and diffuse out enough material that is required for metabolism to occur. So because of that, certain organisms need specialized exchange surfaces and transport systems instead of relying on diffusion like unicellular cells. So explain how the alveoli is adapted for gas exchange by diffusion between air in the lungs and blood in the capillaries. So here you can see that there's oxygen diffusing from the alveoli into the blood capillary from a higher concentration to a lower concentration, down a concentration gradient, and then it's the reverse because there's a higher concentration of carbon dioxide in the blood returning to the lungs or the alveoli. The carbon dioxide diffuses out so we can breathe it out. And this is important because both oxygen and carbon dioxide are involved in aerobic respiration. So in aerobic respiration you've got glucose that reacts with oxygen and that produces carbon dioxide and water, which obviously releases energy from the carbon dioxide being broken down. So how is the alveoli adapted for gas exchange? Well, the alveoli is the last part of the lung where the air reaches so that oxygen can diffuse into the blood capillary and carbon dioxide can diffuse out. So there are 3 different types of adaptation you need to know, specifically the first 2. First, you can see that the alveoli has a large surface area to volume ratio, and that increases the diffusion of gases, whether it's oxygen diffusing in or carbon dioxide diffusing out. from the capillary to the alveoli, or vice versa. And then obviously you can see that the walls of the alveoli and capillary are thin, and usually they're 1 cell thick for short diffusion distance. So the diffusion of oxygen from the alveoli to the capillary, or vice versa for carbon dioxide, is a very short distance, so it happens very quickly. So in the exam you want to mention the wall of the alveoli and capillary. Not a common mistake is to say the alveoli and capillary are 1 cell thick. It's the walls that are 1 cell thick. And then there's also continuous blood flow. So because blood is continuously flowing through the capillary, that means that it's taking away oxygen, and that means that you've always got that oxygen concentration gradient between the alveoli and capillary, which is causing the oxygen to diffuse from the alveoli to capillary. It would take too long for materials to diffuse through cells on the outside of a tissue to reach cells on the inside. So obviously humans can't be one big cell, so that means they have to be multicellular. But the problem with being multicellular is having lots of small cells means the cells that are in the middle would take too long for materials to diffuse to them or to diffuse out. So therefore you need a capillary sort of system where you've got these blood vessels that can fit between these cells, and that means because they're closer to individual cells, they could diffuse useful substances and diffuse waste substances out much quicker. And then this therefore requires a transport system. So we've got the circulatory system in humans that allow this to occur. So how could you increase the rate of diffusion? There are 3 different methods that you need to know about. So here in this example, you can see on the right side there is a bigger difference in concentration. So this means that you could increase the concentration gradient if you want to increase the rate of diffusion. So remember, in diffusion, particles move from a higher concentration to a lower concentration down a concentration gradient. Remember, a concentration gradient is a difference between 2 concentrations in 2 different areas. So having a greater difference between those concentrations means that there's a faster rate of diffusion. So that's one way of increasing the rate of diffusion, increase the concentration gradient. Another way to increase the rate of diffusion is to shorten the distance. So you can see on the right side there's a shorter distance. That means the rate of diffusion is going to be a lot more faster because it's a shorter distance. And then a third method of increasing the rate of diffusion is to increase the surface area, and that means that there is more surface for the substance or particles to diffuse across. So you can see here on the top there's a increase in surface area, so there's more surface for diffusion to occur. And so the larger the surface area, the faster the rate of diffusion. So these are the 3 different variables in terms of how they affect diffusion. A concentration difference, because of which creates a concentration gradient. The greater the difference, the faster the rate of diffusion. The surface area, if there's a greater surface area, that means the rate of diffusion is faster. And then in terms of membrane thickness or distance, the greater the distance or the thicker the membrane, the slower the rate of diffusion. So this one's different to the other 2. It's the opposite. If the distance increases, the rate of diffusion decreases. So if we go back to concentration difference, which creates a concentration gradient, you could see that when there's a greater difference between 2 concentrations, the rate of diffusion increases. And we could say that the rate of diffusion is proportional to the concentration difference. In other words, if we double the concentration difference, the rate of diffusion also doubles. So if we had 2 different areas where there has 2 different concentrations, and we were to make the concentration difference twice as large, the rate of diffusion would be also twice as fast. Now the rate of diffusion is also proportional to surface area. So again, if you double the surface area, the rate of diffusion will also double. However, when it comes to distance, the further particles have to diffuse, the slower the rate of diffusion. So in this case, it's due to the thickness of the membrane. If you increase the distance, that means you're actually decreasing the rate of diffusion. And so this is an inversely proportional relationship where distance increase causes a decrease in the rate of diffusion. So in this example, you can see here, if we were to double the thickness of the membrane, in other words double the distance, the rate of diffusion will now be, because it's the inverse, 1/2. So it will halve. So doubling the distance meant that the rate of diffusion halved. So this isn't directly proportional, it's inversely proportional. Now we could take into account all of those three variables and look at the relationship using a calculation called Fick's law. So in Fick's law, the rate of diffusion is proportional to, we already know, the surface area and the concentration difference. However, it is inversely proportional to the thickness of the membrane. So those three relate to each other in terms of how they're proportional to the rate of diffusion. So if you look at an example here in question 8, the carbon dioxide diffuses into an alveolus at 0.0001 grams per second. So calculate the rate of diffusion. If A, the surface area is reduced by 30%, so if we reduce the surface area, we should understand that the rate of diffusion should also decrease by 30%. So if the rate of diffusion was already 0.0001 grams per second, and it's now going to reduce by 30%, that means it is now going to be 70% of what it was before. So in other words, it's just 0.7 times what the rate was previously, because rate of diffusion and surface area are directly proportional. So if the surface area is reduced by 30%, the rate of diffusion should also reduce by 30%. Okay, in question 8b, the concentration difference is increased by 20%. So if you increase by something by 20%, it is now 1.2 times more than what it was. So if the concentration difference increases by 20%, the rate of diffusion should also, because it's directly proportional, increase by 20%. So it's 1.2 times the value it was previously. And then if we look at 8c, the alveolus wall thickness is halved. So this time round, if you halve the thickness, that means you're halving the distance the gases have to diffuse across. So if you halve the distance, we know that the rate of diffusion is inversely proportional to the thickness of the membrane. So in this case, if you're halving the distance, the rate of diffusion doesn't halve, it actually increases. So how much will it increase by? So we need to use the formula, the inverse formula. So it's 1/2, and 1/2 is 2. So in other words, the rate of diffusion will double what it was previously. So if the thickness is halved, the rate of diffusion will double because it's 1/2. So that's because it's an inversely proportional relationship. So you can see in this question here, using Fick's law, all three of these have an impact on the rate of diffusion. Two of them have an impact where it's directly proportional. So when they increase by a certain factor, the rate of diffusion will also increase by a certain factor. So that's surface area and concentration difference. However, the thickness of the membrane is inversely proportional. So when it increases by a factor, the rate of diffusion will increase by the inverse of that factor. In other words, it actually decreases. And you should be able to then do calculations based on what they ask you in terms of what increases, what decreases. So these are all the things you should know based on the spec from the information that we've just discussed. Explain how the structure of the blood is related to its function. So first we need to know what the structure of the blood is, what is in the blood. So the blood contains plasma, that's the liquid part of the blood. It also contains white blood cells, and there are two different types of white blood cells you should know, which you should remember from the previous unit, SB5. So they're phagocytes and lymphocytes. They also contain platelets, which are fragments of cells, and then the last one is obviously red blood cells, which is scientifically known as erythrocytes. And we want to know how the structure of these blood components is related to its function. So we're going to know their function as well and then relate that to its structure. So first start off with, we've got red blood cells, and the function of red blood cells is to transport oxygen from the lungs to the body cells. So as we've discussed earlier, the red blood cells transport oxygen from the alveolus to the body cells. So how are they adapted? So these are the three adaptations to mainly know about. First of all, you can see that they've got this sort of disc shape, which is known as biconcave. And because of the specific shape it has, that increases the surface area to volume ratio, which makes it easy for gases to diffuse into the red blood cells, which would be oxygen, and it makes it easy for oxygen to also diffuse out of the red blood cells when it needs to get to the body cells. So having a biconcave disc shape creates a larger surface area to volume ratio for gas diffusion in and out of the red blood cell, so that it can transport oxygen to body cells. And then another adaptation is the fact that red blood cells contain hemoglobin, and so hemoglobin is the pigment that binds and carries oxygen and then converts to oxyhemoglobin. So in this adaptation, there's a pigment, red pigment, that it contains, and because it can bind onto oxygen that diffuses into the red blood cells, it can then transport that. When the red blood cells transport through the blood vessels to the body cells, the oxygen would be binded to hemoglobin in the red blood cells, so it could transport it to the body cells. And then if the hemoglobin is the pigment that's going to transport it within the red blood cells, you want a lot of these hemoglobin to be in the red blood cells. So another adaptation is that the fact that the red blood cells have no nucleus, and because they have no nucleus, that means there's more space for more hemoglobin to be in the red blood cells. And if there's more hemoglobin, that means these red blood cells can carry more oxygen to the body cells. So those are the 3 adaptations that you should know about. And then if you look at the other blood components, there's blood plasma, which is the liquid part of the blood, and it obviously carries the blood cells, which is the red blood cells, white blood cells, and also the platelets, and transports them to the through the blood vessels. Then the blood plasma, because it's a liquid, it also contains dissolved substances, so things like carbon dioxide, which is produced by the body cells after respiration. The majority of the carbon dioxide is actually dissolved in the blood plasma, and that's what makes it acidic, unfortunately. So that is transported by the plasma. And then there are other useful substances such as glucose that could also be transported in the blood plasma to the body cells. And then we've got white blood cells, and we know that white blood cells are part of the immune system, so they attack pathogens in the body, and there's 2 different types you need to know about. So we've got phagocytes, and these phagocytes, they ingest, or in other words, they engulf the pathogens and destroy them using their lysozyme enzymes that they have inside them. And then we've got another type of white blood cells, which are known as lymphocytes, and then lymphocytes are the white blood cells that produce specific antibodies that attach to the pathogens and then destroy them. So they're attached to the antigens of the pathogens, which we learnt in SB5. And then to finish off with, the last blood component we need to know about is platelets, and these platelets are just fragments of cells, and what they do is they help the blood to clot, and therefore if a blood vessel is being damaged or if the skin's being cut, then these clots can block the wound and they prevent pathogens getting into the blood. So that is their function, is mainly to cause the blood to clot. Okay, specification 8.7, explain how the structure of the blood vessels is related to the function. So again, this time around we want to know about the blood vessels. So you can see here is a blood circulatory system, and there's different types of blood vessels we should be able to spot. So you've got some veins here and some arteries, and there are also capillaries. So the different blood vessels have different jobs. So first we want to identify them. So the blood vessel that carries blood away from the heart is known as the artery. So because it's away, it's the artery. Then we've got the blood vessels that are in the body cells, and these are known as capillaries. So that's where substances from the blood diffuse into the body cells, and then waste substances can diffuse out back into the blood vessels. It happens through the capillaries. And then carries blood back into the heart. These are the veins. So if it's into the heart, it's vein. So that's one way of remembering it. Artery, away from the heart, vein, into the heart. And so here are the adaptations of these 3 different types of blood vessels. So you can use the diagram on the left hand side to have a look at what the blood vessels look like. And you can see in the artery, we said earlier, it takes oxygenated blood away from the heart, unless it's the pulmonary artery, which we'll talk about later on. And if you look at the artery blood vessel, which is in the middle of this slide, you could see that it's got thick wall. And that thick wall is composed of strong muscle and stretchy elastic layers. And the strong muscle and stretchy elastic layers help the blood vessels withstand high blood pressure. So because this is blood that is being carried away from the heart, that means there's a really high blood pressure because there's more collisions of the blood with the blood vessel due to the strong contraction of the ventricles. So this means that there'll be a large blood pressure or pressure on the blood vessels. So to withstand that pressure, the arteries need really thick wall, and that thick wall is composed mainly of muscles which are really strong, so thick muscles, and also composed of a thick elastic layer which stretches when the blood collides, and that withstands the high blood pressure because it means that the blood vessel can stretch, which increases its volume, and that will then decrease the pressure. Then we've got capillaries. As we said earlier, capillaries are the where exchange of substances happen in the body cells. So they're in between artery and veins, and they're found in the body cells. So substances such as oxygen and glucose can diffuse into body cells, and carbon dioxide can diffuse out. And if you look at the bottom on the picture of the capillaries, they are thin, or they're one cell thick in terms of their wall. So not the capillary, the wall of the capillary is one cell thick. And they also have a narrow lumen. And this means that when the blood flows through it, specifically the red blood cells, there's a short diffusion distance. So for things to diffuse, or substances to diffuse from the capillary to the body cells, the distance that it has to travel is very short due to the fact that the lumen, the hole, is very narrow, and also the fact that the wall of the capillary is only one cell thick. So it makes it quicker for substances to diffuse out, and it's also quicker for substances to diffuse in. So things like waste substances such as carbon dioxide and urea. And then if the substances are going to diffuse in and out, those walls have to be permeable for diffusion. So capillaries have got walls that are permeable that allow the substances to diffuse in and out. So that's another adaptation that they have. And then to finish off with, we've got the last blood vessel, which is veins. And remember, these are the ones that take deoxygenated blood into the heart, with the exception of the pulmonary vein. And because the blood pressure is much lower once the blood has travelled around the body and is returning back to the heart, this means that there's a problem because the blood is travelling slowly, and that means that sometimes it can flow backwards. So to prevent backflow of blood, the veins have got valves in them, and those valves prevent backflow. Also, because there's not a lot of high blood pressure, that means that their walls don't need to be very thick. So they have thinner walls in comparison to the arteries. And you can see that on the picture on the far left. You can see that the walls are a bit more thinner than the artery, and they've got a more wider tube or wider lumen. And here you can see a picture of electron microscope picture of both an artery and a vein, and you should be able to spot that the one on the left is an artery because the wall is a lot more thicker and it's got a more narrow lumen because of that, in comparison to the one on the right, which is a vein. It's a lot more thinner wall, and it's got a wider lumen. Okay, specification 8.8, explain how the structure of the heart and the circulatory system is related to its function, including the role of the major blood vessels. So that means we know those blood vessels, but they've got specific names, the ones that are close to the heart. And then we need to know about the role of the valves and the relative thickness of the chamber walls as well. So first of all, when we look at the structure of the heart, we should identify that there are actually 4 chambers. So the chambers in the top are known as atrium, and the chambers at the bottom are known as ventricles. And you can see the heart is split into two as well, in terms of left and right. So the left side of the heart is where oxygenated blood is carried into, and then it is pumped around the body. And the right side of the heart is where deoxygenated blood arrives in the heart, and that is then pumped to the lungs. And blood always transports into the heart through the atrium and leaves the heart through the ventricle. And when it transports in and out, then it has to go through blood vessels. So we're going to know about blood vessels. There are obviously two blood vessels, one going from the heart to the lungs and one going from the lungs to the heart. And then you've got another two blood vessel where it's from the heart to the rest of the body, and then from the rest of the body back to the heart. So there are four blood vessels altogether. The two that are related to the lungs are known as pulmonary. So we've got pulmonary arteries and veins. So remember, arteries take, in this case here, they take blood away from the heart. So away from the heart and to the lung is the pulmonary artery, and then veins is into the heart. So the pulmonary veins takes blood from the lung, which is pulmonary, and into the heart. And then you got two other blood vessels that you need to know about, the aorta and the vena cava. And the aorta is an artery, so this means that it's a blood vessel that takes blood away from the heart. So this is taking it away from the heart to the rest of the body, because we already know that the pulmonary artery takes it to the lungs. And then we've got the vena cava, which returns deoxygenated blood back to the heart. And then there are also valves that we need to know about. So between the atrium and the ventricles, we have got two different type valves called bicuspid valve and tricuspid. So on the left side of the heart is the bicuspid, and the right side of the heart is the tricuspid. And notice when we're looking at the picture, when I say that the left side of the heart, we're actually looking at the picture that is on the right side. So you have to imagine that you're standing on the opposite side of this slide, and the bicuspid is on the left and the tricuspid is on the right. And then when the blood goes away from the heart, it goes through valves known as semilunar valves. Okay, so we already know in a respiration, oxygen and carbon dioxide has to diffuse in and out of the alveoli so that we can get oxygenated blood and we can diffuse out the carbon dioxide. And obviously those gases are then involved in body cells where the oxygen is used for respiration and the waste product is carbon dioxide. So if we start off from here, we've got oxygen that's diffusing in from a higher concentration in the alveolus to a lower concentration in the blood, and this is the capillary in the lungs, and then that needs to then go to the heart, so the heart could then pump it to the rest of the body, because there isn't obviously a lot of pressure from the lungs, so we need the contraction of the heart to pump it to the rest of the body. And as we said earlier, this is oxygenated blood going from the lungs to the heart, and because it's carrying blood to the heart, or it's going into the heart, it's a vein, and because it's from the lungs, it's pulmonary. So to start off with, we've got oxygenated blood going into the heart through the pulmonary veins, and then it has to go to one of the top chambers. So this is the left side of the heart, so remember it's a mirror image to the way we're seeing it. And this left side of the heart, the top chambers are always called atrium, so the blood goes from the pulmonary vein into the left atrium. And then it needs to go from the left atrium to the left ventricle, but it needs to go through a valve. And the left side, the valve between the atrium and ventricle, is known as the bicuspid valve. And the blood then travels to the left ventricle. And then, as we said earlier, to leave the heart, it needs to go through another valve. And this valve is known as the semilunar valve. And then it's going out of the heart. So away from the heart, those blood vessels are called arteries. And this artery has got its own specific name. It's known as the aorta. So it's taking blood away from the heart and to the body cells. And then the artery takes the oxygenated blood to the body cells. And when it reaches the body cells, we've got a different type of blood vessel. We've got the capillaries, which we mentioned earlier. So remember, these are very narrow blood vessels, so that it means that they can fit in between lots of individual cells, so it makes the diffusion distance a lot more shorter, and that increases the rate of diffusion. So oxygen diffuses out of the red blood cells, and then we've got carbon dioxide diffusing from the body cells because of respiration. It's a waste product, and the blood then carries the carbon dioxide and needs to be transported to the lungs. And while this is happening, the pressure is decreasing as the blood is traveling along, so that means that it needs to go to the heart first, so the heart can pump it to the lungs. And when it goes into the heart, this is a vein, and this vein has got its own specific name. It's known as the vena cava. And then when it goes into the heart, this is on the right side of the heart. This is deoxygenated blood, and because it's deoxygenated blood going on the right side of the heart, we know the top chamber is always called atrium, and it's the right side, so it's right atrium. And then it needs to go from the right atrium to the bottom chamber, which is the right ventricle, but it must go through a valve, and this time it's not the bicuspid valve. This is known as the tricuspid valve. Goes to the right ventricle, and then for blood to leave the heart, obviously it's carrying blood away from the heart, so it's an artery and it's going to the lungs, so it will be pulmonary artery, but it has to go through a valve first to leave the heart. Blood that leaves the heart always goes through a valve known as cellular valve, and then obviously when it's going to go to the lungs, it's going to go through the pulmonary artery, as I've just mentioned. And then the deoxygenated blood goes to the pulmonary artery, the carbon dioxide diffuses out into the alveoli, and then obviously the blood then takes oxygen, which diffuses in, and we go through that whole circulation again. And you can see this is a double circulation, because there's one circulation from the heart to the body cell, and there's another circulation from the heart to the lungs. So there's two circulations. So humans have got a double circulatory system. So there are additional things we need to know. These are the roles of the major blood vessels we discussed, but now we need to discuss the role of the valves. And just like the veins, the valves do the same job in the heart, so they prevent the backflow of blood. So we've got both the bicuspid, tricuspid and semilunar valves, all four of them in the heart, make sure or prevent the blood from flowing in the wrong direction. And then we also need to know about the relative thickness of the chamber walls. So here, the wall of the left ventricle contains more muscle than the right ventricle, right? And the ventricles also contain a thicker wall than the atrium, the top chambers. So why is the left ventricle the thickest wall? Because we know it contains the most muscles, and the reason why the left ventricle is got the thickest wall and contains the most muscle is because that is the chamber that needs to contract to pump blood to the rest of the body. So the left ventricle muscle cells, and there's more muscle cells in the left ventricle wall than in the other chambers, they have to contract more stronger to pump blood around the whole body, and because they have to pump blood around the whole body, while the right ventricle only needs to pump to the lungs, they pump it under a higher pressure so that the blood can reach those cells that are further away in the rest of the body. So that's why there needs to be more muscle cells in the left ventricle, because the blood needs to be pumped under a higher pressure, and because it needs to be pumped under high pressure, that means that the blood can then reach cells that are further away around the whole body, and that also helps to withstand some of the pressure as well. So having a thick wall means that it could contract stronger because there are more muscle cells there. And then obviously the right ventricle has a thinner wall in comparison to the left ventricle, because it only needs to pump blood to the lungs, which is a very close distance. And then the atriums have really a lot more thinner walls in comparison to the ventricles, because they only contract and pump blood to the ventricle, which is just nearby, so they don't need to have very thick walls in comparison. So the thickest wall is found on the left ventricle because it needs to pump blood around the whole body. There's some additional information about the heart which we just discussed. And then this is the spec, just to finish off, just describing the content that we just went through. And then to finish off, one more topic about the heart. We should be able to calculate the heart rate, stroke volume and the cardiac output based on specification 8.12. So in this case, the contraction and relaxation of muscle cells during each heartbeat is controlled by impulses in the nervous system. We know that the heart rate is how often the heart beats in a minute. So if your heart beats, say, 60 times in a minute, that means your heart rate is 60 beats per minute. Now the volume of blood pushed into the aorta in each beat is the stroke volume. So the stroke volume is how much blood, the volume of that blood, pushes out of the left ventricle into the aorta every beat. And if we multiply that by the amount of beats in a minute, we can then work out the cardiac output, which is the amount of blood that is pushed out of the left ventricle and into the aorta every minute. So heart rate is how many times the heart beats per minute. The stroke volume is how many litre of the volume of blood that is pumped by the contraction of the left ventricle to the aorta in one beat. And if we multiply those two together, obviously the volume multiplied by how many times it beats in a minute will then give us a total volume in a minute, which is known as the cardiac output. So to work out the cardiac output in the exam question, this cardiac output equals stroke volume multiplied by heart rate. And if you exercise regularly, regular exercise increases the strength of the heart muscles and the ventricle size. So in other words, if you're someone who's a lot more fitter, they tend to have a bigger stroke volume. And if you have a bigger stroke volume, but your cardiac output tends to be the same as someone who doesn't exercise, that means that you generally have a lower heart rate. And that's why people who are a lot more fitter generally have a lower heart rate, because their stroke volume is actually a lot more greater. They could actually pump more blood from the left ventricle into the aorta every time the heart beats, and that's because they've got a stronger heart muscle and ventricle size is a lot bigger, so they could therefore pump more, and therefore their heart rate could actually be a lot less. So if you look at some examples here, the heart rate multiplied by the stroke volume equals the cardiac output, and you can see examples of calculations. If the heart rate is 60, the stroke volume is 0.06 litres per beat. Just multiply them together, you get 3.6 litres per minute. Moving on to our last topic, cellular respiration. Specification 8.9 describes cellular respiration as an exothermic reaction which occurs continuously in living cells to release energy for metabolic processes, including aerobic and anaerobic respiration. So in cells, there are two different types of respiration that could happen. In cellular respiration is when you're talking about both types of aerobic and anaerobic respiration. So respiration happening in cells releases energy from glucose, and because it releases energy, some of that energy heats up the body and is lost to the surrounding. So we can say that it's an exothermic reaction. And the main type of cellular respiration is aerobic respiration, and this needs oxygen. And because it needs oxygen, that reaction mainly occurs in the mitochondria. And we should know the word equation. Therefore, obviously the energy is released from glucose and it needs oxygen, so glucose reacts with oxygen, and the byproducts or the waste products are carbon dioxide and water. And because it's an exothermic reaction, that releases energy, and that energy can then be used for other cell processes or metabolic reactions. So even simple things such as movement, but then also it could be driving other reactions that happen in the body, such as protein synthesis, or synthesizing other molecules as well, or breaking down molecules as well. However, during strenuous exercise, oxygen is used up faster than it's replaced. So in this case, you're starting to lack oxygen. So you're breathing in oxygen, but not enough oxygen is going to the body cells to release energy from glucose. So you continue to do aerobic respiration, but then anaerobic respiration can also occur in the cells, and they can be also used to release energy from glucose. So anaerobic respiration is also an exothermic reaction, but in this case no oxygen is required, but the problem is lactic acid is produced. So if you look at the word equation, there's no oxygen, so it's just glucose on its own, and when you break down glucose you produce lactic acid. So specification 8.10, you've got to compare the process of aerobic respiration with the process of anaerobic respiration. So here I've written the differences, because compare means either tell me the similarities or the differences. We already know the similarities. They're both reactions that release energy from glucose, and they're exothermic reactions. So what are the differences? So you can see here, aerobic respiration, glucose reacts with oxygen to produce carbon dioxide and water. By anaerobic respiration, we only have glucose as a reactant, there's no oxygen, and the product is different as well. So although both release energy, in anaerobic respiration the physical matter product that is produced is lactic acid, whereas in aerobic respiration is carbon dioxide and water. And as I stated, anaerobic respiration doesn't require oxygen and it produces lactic acid, but the problem with the lactic acid, because it's acidic, it makes muscles tire a lot more quicker, and that's known as muscle fatigue. That's because it's affecting the enzymes in the muscles, specifically the ones involved in respiration, and because it's affecting them, that means there is muscle fatigue occurring. So we're going to get rid of the lactic acid, and to get rid of the lactic acid you have to breathe in extra oxygen. So after you've stopped exercising, you'll notice that your breathing rate is still very high, and that's because the extra oxygen is being used to break down lactic acid into carbon dioxide and water. So lactic acid that is built up, which cause muscle fatigue in muscle cells, they need to be broken down by the extra oxygen, and this is known as oxygen depth or epoch, and that oxygen depth is the amount of oxygen required to break down lactic acid into carbon dioxide and water after exercise has been finished. Now, one big difference between aerobic and anaerobic respiration is the fact that aerobic respiration releases a lot more energy than anaerobic respiration, and also because it requires oxygen, most of the reaction happens in the mitochondria, whereas anaerobic respiration only occurs in the cytoplasm. So if you release more energy doing aerobic respiration, what's the advantage of doing anaerobic respiration? What's the point? Why not just do aerobic respiration all the time? So the big advantage of anaerobic respiration is the fact that you could release a quick short burst of energy without any presence of oxygen. So if you're lacking oxygen and you still need energy, then you could have a really quick short burst of energy released from the glucose without there being any oxygen. However, the disadvantage, as we mentioned, is it produces lactic acid, and because it produces lactic acid, that can cause muscle fatigue. So that means after you finish exercising, you still breathe very heavily, because that extra oxygen that you breathe heavily in for the oxygen depth, the epoch, that extra oxygen is required to break down the lactic acid into carbon dioxide and water. And then the other big disadvantage of anaerobic respiration is, even though it provides a quick short burst of energy, you can't release energy for long term. So it releases a lot less energy than aerobic respiration. So here you can see there's a few additional information about muscle fatigue and how the pH affects the enzymes in muscles, and then obviously the oxygen required to break down the lactic acid afterwards is known as oxygen depth. And this is a 6-mark question. Compare and contrast the use of aerobic and anaerobic respiration in marathon racing, which you can read. Now, fungi and plants can also respire anaerobically. However, when they respire anaerobically, they don't produce lactic acid. So they could do anaerobic respiration, which is respiration where energy is released from glucose but without the use or presence of oxygen. But instead, their products are ethanol and carbon dioxide. And ethanol is obviously used to make alcohol, so that happens through anaerobic respiration. And then also when you're baking, When the bread rises, that's because of the carbon dioxide produced as well, so that involves anaerobic respiration. So both yeast, fungi and plants can also respire anaerobically as well as respiring aerobically. And then here's just a summary of the specification to finish off with. So we've already talked about the two types of cellular respiration and the fact that they're exothermic, and we've compared and contrasted both aerobic and anaerobic respiration. So to finish off with, we've got specification 8.11, which is the core practical, where you investigate the rate of respiration in living organisms. So you want to see how organisms respire differently in different conditions, what makes it faster or slower. And to do that, you have to measure respiration using an equipment called a respirometer. And the respirometer measures the amount of oxygen that was used up. So what you do is you have a tube that you put your organism into. It could be wood lice or any other sort of small organisms, and then when you use those organisms, you obviously have a fixed volume in that tube. You connect it to some sort of a sort of colored liquid in a capillary tube, and then that liquid should move based on the fact that the organisms are using up oxygen for respiration. So when they're taking in the oxygen, if they're doing more respiration, they'll be taking in more oxygen, and that will decrease the volume of the air. But we know in respiration that the organisms release carbon dioxide, so that should increase the air. But the reason why the air volume doesn't increase when they release carbon dioxide is because we add something called soda lime, and that soda lime absorbs the carbon dioxide. So it doesn't matter if the organism is doing more or less respiration, it's going to be absorbed by the soda lime. So any changes in the volume of the air will be due to how much oxygen is being used up. So if there's a faster rate of respiration, that means more oxygen would have been used up. So that means the volume would have decreased, and the blob of liquid in the capillary tube will move towards the organism, and the rate that it moves can then be calculated. So in other words, it's the volume of oxygen that's taken up divided by how long you time it, and if that calculated rate is faster, in other words, if the liquid moves quicker, that means or it moves a bigger distance in a certain amount of time, then that means the rate of respiration was a lot more faster, because the organisms used up more oxygen, and that decreased the volume. So you can see here, in this case, the independent variable would be the type of organism in investigation 2 if you're comparing different types of organism, or you could look at temperature, because temperature has an effect on respiration because of the fact that it affects the enzymes involved in respiration. And then the dependent variable would be the distance that the blob of colored liquid moved, or in other words, the speed or the rate. So if you divide it by the time. And then you got control variables. So every time you're doing control variable, you got to make sure that the mass of the organism is the same, not necessarily the number, because you could have some organisms that are larger or smaller. So you want to make sure the mass is the same. And then obviously, depending on what you're investigating, if you're investigating temperature, then you want to make sure that you're using the same type of organism. In other words, if you use wood lice at say 30 degrees Celsius, and then you change the temperature to 20 degrees Celsius, you want to use the same type of organism. If you're comparing different types of organism, like wood lice with say a different type of organism like maggots or something else, then you want to make sure that the temperature is constant and it's controlled. And then you could also do a control experiment to check that your independent variable is the thing that is having an effect on the movement of the colored liquid. And to do that, if you're checking the type of organism, what you could do is you could either put no organism in the tube, or you could put a dead organism in the tube, or in this case here in this picture you can see that they put glass beads, although in the exam I would prefer you've written no organism or dead organism, and therefore there'd be no respiration. So that means the colored liquid will not move. Now, risk assessment is a popular question for this practical, because you're using soda lime that is corrosive. You want to make sure that you're not in contact with it, and also the organism is not in contact with it, because you've got to think ethically about the organism. So you use a cotton wool that acts as a barrier to prevent you touching the corrosive soda lime or the organism touching it as well. And then the other thing you could obviously use is safety goggles or gloves to protect yourself from the corrosive soda lime. Then ethical consideration, you have to make sure that you release these organisms after you finish the experiment. But the other thing to watch out for is if you're doing temperature, you can't use a temperature too low because then it could harm the cells of the organisms. But it can't be too high above the optimum temperature either, because then the enzymes in the organism could denature and that could kill the organism. So you want to use temperatures that the organism experiences normally in nature, so you can't do it temperatures too low or too high. And again, ethical consideration comes more into play for this practical than any other practical. So if they ask about ethical consideration, it will be more likely for respiration rate practicals.